Hello everyone, my name is Daniel Bogush and I'll be presenting on behalf of Rivendell Therapeutics, a company that I founded with my PI and mentor, Dr. Sinisa Dovat, and my other mentor and our chemist, Dr. Demonte. The therapeutic efficiency of topical Poly INflammatory Kinases, or PINK inhibitors, in preclinical models of psoriasis and irritant contact dermatitis. So the current problem is that 7.5 million people in the United States have psoriasis, and 80 to 90% of them have mild to moderate instances of this disease. And within that, there is also irritant contact dermatitis, which accounts for about 80% of all contact dermatitis in all of skin disease. And it's estimated that 30 to 50 million people in the United States suffer from some form of skin inflammation. And this is mirrored within Europe as well. So kinases are enzymes which are often overexpressed in malignant diseases in humans. And many kinases are pro-inflammatory. Now, inhibition of these kinases using systemic kinase inhibitors show improvement in several inflammatory conditions, such as Crohn's disease. However, currently, there are very few systemic kinases that are used and have been tested in phase 1 and phase 2 trials for various malignancies and inflammatory diseases. So our goal was to develop a kinase inhibitor for topical usage against these skin inflammation diseases. The market size for this, and a part of the reason that we are looking into dermatology, is that patients with psoriasis account for about 24 million people in the United States. Now, patients using topical treatments to address this are about 16.3 million. And then irritant contact dermatitis patients using prescription treatment account for about 10.6 million people. And the annual out-of-pocket expense is exceeding $500 per person that is using this. So we come primarily from a leukemia background. Our practice and our lab studies leukemia and anti-cancer drugs and small molecules. That's what we attempt to develop. However, we are still scientists. So when we make a molecule that has really good anti-inflammatory properties, we don't just stop at cancer. We see what can we use that molecule for. And that is how we ended up in dermatology. And we coined these PINK inhibitors. Here is a kinase tree. So we did a protein kinase assay on our lead compound, DDL. Now, let's see if this, okay. So in most kinase trees, when you're looking at inhibition of a specific kinase, it will usually target one pathway of this tree, and it'll hit it about 10-fold. However, when we looked at ours, surprisingly, it didn't hit one target on a kinase tree, it hit a multitude of them. And instead of hitting it 10-fold in a single area, it hit it between 3 and 4-fold in just about every area. This we've never seen before, and we've never seen a small molecule on the market that is currently able to do something like this. And that's why we decided to proceed with testing of this small molecule. So the first thing we did, not being specifically a dermatology lab, is can we test it in dermatological diseases? And the first bar graph shows that yes, we can induce psoriasis. We can induce it in both severe and mild to moderate. And when we apply our small molecule to those diseases, we did see a reduction in the inflammation that it produced. Now, after that, we went on to irritant contact dermatitis. Did very similar study, saw if we could induce the disease state, and then we applied our small molecule, and we did see a reduction in the inflammation that it was produced. And I know a lot of the time, people give me flack for putting everything in black and white and not introducing enough color. So here are the microscopy slides that one of our colleagues who primarily works with skin diseases, who assisted us in getting these H&E stains done and doing the analysis of all of the skin thickness, we see that the first series of slides is untreated. The second series shows inflammation of the respective disease, and then DDL, our leading compound. If you compare the second to third picture, there's a decrease in the inflammation between the two. Afterwards, we then looked at the anti-inflammatory effect over all of the cytokine profiling. Now, this was more of a confirmation in what we already knew, but it gave us some more information as to what cytokine profiles this actually hits. And these are also going to be used as our future indications to look outside of just psoriasis and irritant contact dermatitis. So then the question is, if it's hitting all of these kinases, is it safe? And we recently submitted our first SBIR that we did get scored, and we were looking over the reviewer comments, and despite stating several times that we thought it was safe and giving them the data that we produced, they were still very skeptical of that. So here in this slide, we administered DDL both IP and orally over the course of 3 weeks at what we would consider a very high dosage, and the BID, CBC, differential, platelets, electrolytes, kidney function, and liver function tests were all normal. So based on our mouse examination, we believe that it is safe and can be used. We had no adverse effects when we treated the mice. So it is a very good indication moving forward that these are safe. And despite the fact that they do target so many things, because they don't target them as hard as other kinase inhibitors, because we have more of a broad range, we're not seeing any adverse effects. So what does the current market look like? So currently, if you were to go in and you wanted to get prescribed something for your irritant contact dermatitis or psoriasis, the leading indicator is Tapinarof. It costs out of pocket, if you were to go to the pharmacy, about $1,400. In order for us to make ours, we can make it for about 50 bucks. So despite the fact that that price may go up a bit due to marketing or production, making this at scale, we believe that we can keep it well under what the competitors are doing. Now, you might be asking, well, that's great, but how good is your small molecule in compared to Tapinarof? Well, we did a small study looking at exactly that. So we induced our disease state, and then we applied Tapinarof as well as our lead compound, DDL. Tapinarof had about a 20% reduction in ear thickness, whereas DDL had a reduction of about 40%. So we are currently exceeding what is on the market. So in conclusion, we discovered 3 novel first-in-class small molecules that are poly inflammatory kinases. DDL shows strong therapeutic effect in preclinical models on psoriasis and irritant contact dermatitis. And in those preclinical models, the therapeutic effect of the leading compound is superior to what is on the market currently. And then I will reiterate again that there is no toxicity has been observed following topical or high-dose oral administration of the DDL. Our commercialization plan, we are in the middle of acquiring funding for our IND and pre-IND. We have submitted an SBIR, our first submission as a company, and unfortunately, there were some setbacks due to the government shutdown and the NIH reviewing process, which I'm sure many of you have heard about. But we were able to get scored, which is fantastic. And then we are addressing reviewer comments, and pending everything goes well, because they're having their SBIR vote today. Pending everything goes well, we will be submitting on the 5th of March. And then our next steps are formulation, our IND-enabling experiments, mostly moving to larger preclinical models. Nanopigs are the standard in dermatology. And then we're trying to check all of the marks off for human safety tests so that we can eventually move this into clinical trials. Our current business needs include IND-enabling experiments, experienced people with skincare products and pharmaceuticals, experience in processing of FDA approval for skincare drugs, established safety and patient usage, and then marketing, and then how to develop phase 1 and phase 2 clinical trials as well as our IOB approvals. So yeah, thank you so much for your time. I really appreciate it.
Did you file for a patent on this?
Yes, we did. So through Penn State, we actually have a provisional patent currently that is being processed for all of our molecules. We have the patent on dermatological models. We have a patent on the primary compound, as well as quite a few of the compounds that can lead from this with their derivatives. And then we are working on getting patents for additional disease states on top of just dermatological models.
Can you tell us a little bit about the backstory, how you stumbled upon this? The derivation, I assume it's a small molecule. Can you show us the structure or anything of that nature?
So I don't have the structure in these slides. And then because we are still in a provisional patent, we are holding off on releasing the structure until absolutely necessary. However, how we actually came up with this is we have quite a few compounds that we primarily use with our cancer treatments. And we are primarily looking at synergy studies to reduce the toxicity in cancer. We did quite a few derivatives of some of our current small molecules that we're using, and we found one that just had very high anti-inflammatory properties, and we didn't really know what to do with it. So our PI, Dr. Sini Sadovat, he gave us the opportunity to test in a few mice with the full thought being, hey, it's not going to work, we're just going to move on. But it kept working. It worked in every model that we used, and we continued testing. We did cytokine profiling. We did single cell sequencing using RNA. We started doing testing in other model systems and additional disease states, and it showed promise in those as well. And we're currently working on getting approvals to do additional tests in more disease states and looking at collaborators, especially within Penn State, that can allow us to test in additional diseases that also focus or are theorized to have a cause of anti-inflammation.
A lot of those things seem like they were, I'll say, internal, health and safety wise and those kinds of things, as opposed to business and investor wise.
Yes. So our funding that we're currently looking at, so currently we are doing non-dilutive funding through the NIH, and that's where our SBIR was and where it came from. We're in the process of going with that route. We are primarily a scientific lab that has a decent track record of getting grant funding through the NIH, so we figured we would start there. So currently we are still looking for funding to go through our formulation and IND enabling experiments, and we're not going to rule anything out. Ultimately, we are a company that is made up of doctors, and I know that especially the three founders, our aim is to help as many people as possible, and we believe that this can help a lot of people, and we hope that we can get this to them as quickly as possible. So we're not ruling out any mode of funding to assist with that.
One of the reasons that I asked that was based on your presentation when you said, we did this, we researched this, we discovered this. That's great information for investors. Then you need to do that presentation on the why, what, and how, and their buy-in related to that. Some of that information they don't care about to that level. So I just wanted to throw that out there, depending upon your audience and what it is that you're trying to accomplish, for how you might do some of your future slides and presentations by what you're trying to accomplish.
Yes, and thank you very much for that information. It has definitely been mirrored before because I was primarily a scientist, and I am attempting to move and continue that transition from academia into business, into small startups. And we've had a lot of great opportunities with the Innovation Fellowship, which I am a member of, where they're trying to develop me more towards a business side so that I can tailor my presentations to those that I'm presenting to. And I'm trying to move it away from a slide deck of entirely nothing but science and data to making it more accessible to everyone else. But I still do have that in my roots, and I tend to bring it out a little bit too often. So thank you for the criticism.
You have up there $300,000 for formulation, optimization, polymers.
Yes.
I would imagine you would just have this small molecule in an ointment and just spread it on your skin. Why do you need nanopolymers?
So the nanopolymers is actually in relation to the deployment of the small molecule. Depending on the cream that you use, can either enhance, add, or have no additional effects to the small molecule. It also helps with absorption into the skin. So the experiments to, one, make the small molecule work with the ointment that we're attempting to get it into, as well as test it for safety, is what costs the $300,000. And then that needs to be done in a multitude of small animals, which also drives up the cost. And then any modification to the deployment also needs to reflect in the safety standards.
Would you be able to do, I guess, this full, including the nanopig? I guess this would be a phase 2 SBIR, and so that would be your preferred kind of non-dilutive funding route in the near term for these deliverables, these near term?
Yes, the SBIR that we submitted was a phase 1, phase 2 fast track SBIR, which covered both the formulation, optimization, as well as the IND enabling experiments on the mini pigs. And it included both of those current modes. We've talked to quite a few people, and that's kind of the entry into clinical trials, is to show that we can do the IND enabling experiment. It's safe in larger organisms before we can start bolstering up to human trials, which is one day our aim.